Bacillus subtilis strains bearing translational fusions of mismatch repair proteins fused to GFP are streaked out on selective media. The streaked cells are used for fresh inoculum into liquid culture. Cells are grown until OD600 is 0.3 to 0.4, whereupon mismatch-inducing treatments are applied if desired, and cells grown further until reaching OD600 of 0.5 to 0.8. Cells are removed, the membrane stain FM464 is added, if desired, and cells are applied to slides with prepared agarose pads and allowed to settle. After removal of excess media by aspiration, a cover slip is applied and cells are visualized by microscopy.
Hi, I'm Dr. Andrew Klocko from the lab of Dr. Lyle Simmons in the Department of Molecular, Cellular, and Developmental Biology at the University of Michigan, Ann Arbor. Today, we will show you a procedure used to visualize translational fusions of the DNA mismatch repair proteins as well as other repair and reposomal proteins in Bacillus subtilis. We use this procedure to study the interaction between the DNA mismatch repair proteins and the reposomal proteins in Bacillus subtilis live cells. So let's get started.
To begin this procedure, prepare the B subtilis strains containing the translational fusion protein you wish to visualize. For some strains, starting 1 day prior to imaging is sufficient. Examples of these include strains that grow well when the translational fusion between GFP and the gene of interest is incorporated into the gene's endogenous locus, such as mut-L-GFP. When starting 1 day prior to imaging, use a sterile stick under sterile conditions to apply the bacteria to selective LB agar medium on plates in order to grow single colonies. Incubate overnight at 30 degrees Celsius. The next day, the colonies will be used for a starter inoculum.